I've covered most of those points. Okay, so the last system I want to talk about today is the type 1 secretion system. So sorry that we're giving you a trip around the zoo of secretion systems, but in previous years we gave a separate lecture to each one of these, but we decided that was a bit over the top this year. So type 1 secretion, here we have 3 components to this type 1 secretion system. So this system is spanning the whole cell envelope. There's an ABC transporter located in the inner membrane. So this is a class of transporters. ABC stands for ATP binding cassette. So these mediate not just protein secretion, but they mediate the translocation of all sorts of molecules, peptides and other small molecules also going through them. So the ABC transporter that is energizing the process through the hydrolysis of ATP. There's then this thing called the membrane fusion protein, a trimeric protein, kind of acts as an adapter, and it connects the things that are in the cytoplasmic membrane and things in the outer membrane. And then there's an outer membrane protein, the Tol-C. Tol-C is the kind of archetype of this group of proteins, but there's actually a wide variety of different members of the Tol-C family involved in type 1 secretion. They form trimers in the outer membrane. And these systems, they don't just export these Tol-C proteins. They're quite promiscuous in what they export. They can export protein toxins. They can also start exporting antibacterial substances and all sorts. So just drawing this graphically, this is what the whole thing looks like. You've got these three different components, three different subunits combining together to make a pore, a channel that connects the cytoplasm all the way through to the outer membrane. So we've got this pore in the outer membrane here, this 12-stranded beta barrel. So the vast majority of proteins in the outer membrane, in fact, are beta barrel proteins, form this characteristic fold. And this Tol-C beta barrel is similar to what we see in another class of proteins called porins, which are also involved in movement of material across the outer membrane. Then projecting down from there, you have all these alpha helices that are sticking out down into the periplasm. So the beta barrel tethering it in the outer membrane, and then these alpha helices dropping down below. And Koronakis is the key name associated with this. A guy who works in Cambridge solved all these structures. And we have a guy called Vasily Bavro, who's actually joining us in the next few weeks, who's also an expert on these systems and the structural biology of them. So what happens with these systems is you have this membrane inner complex, the ABC transporter, and the MFP contacting the outer membrane protein as the structure assembles. You get substrate recognition. So here we have instead of an N-terminal signal sequence, we have a C-terminal signal sequence. So different from what you see with the sec-dependent system. The C-terminal signal, and the C-terminal signal is not cleaved off either. There's no equivalent of a signal peptidase here. As with the sec-dependent system, though, we have these proteins remaining unfolded as they go through the system. So they have to be unfolded to get through the pore. So the MFP contacts with the outer membrane protein, you get this conformational change, and you get formation of this complex, this transient complex. And it's ATP driving all this through. There may also be a role for the protein motive force in driving proteins through the system. There are quite a lot of different sorts of proteins that go through type 1 secretion systems. There is a whole family called the RTX proteins, which are commonly toxins, and they have this 9 amino acid repeat sequence. It's rich in glycines and aspartates, and G's and D's in single letter code. These are very, very long proteins, often over 1,000 amino acids long, and this whole family of them go through the type 1 secretion system. There are some things which are not toxins. So we have these leukotoxins, but we also have proteases, lipases, and so forth. And we have S-layer proteins and things involved in ion scavenging also going through the system. And then I've now finished for today. So, as I say, Julia gave me these slides. I've used most of her slides. There's a series of papers here, review articles, which you can look at if you want to go into more detail on this subject. And I shall put these onto WebCT in the next few days.